My talk is about extracting antimicrobial molecules from the Arctic Ocean. And as I said, there is a dry pipeline, but there is a need for new antibiotics. But I think that's part of the solution. There are many approaches, this One Health, that we need to address to really contribute to this crisis that are upcoming. So I have the pleasure of representing the DigiBiotics-project, and I will walk you through some of how we work with this and inform you about the project and some of the part of some of the results that we have achieved. So let me see. I will see if I can move the slides. Okay, yes. So where do we have the inspiration for why should we look? Are there any drugs coming from the sea? And here I took 3 examples, like marine derived drugs. So if you heard about the snail, where you have this extracted this molecule that is important for chronic pain. This picture in the middle, this tunicate animal have given rise to a soft tissue sarcoma medicine. And the sponge on the yellow one here on the side has given rise to the starting point for developing a breast cancer medicine, Helaven. So there are already medicines coming from the sea on the market. And if I look through the status, there is actually 14 that are approved that are coming from the ocean originally. And there are 23 that are in active clinical trials. When looking at all the medicine, this is a very small part of all the medicines that we have, but it's starting to grow. When I started this some years ago, it was much less, so it has increased. But my concern here is actually that none of these that are actually in the clinical trial or on the market are against antibacterial drugs. So there are no new antibiotics in the pipeline coming from the sea. But luckily, there are many that are in the preclinical pipeline. But as you know, there is a challenge to bring them all the way, because we need to look for new chemistry that could be taken further, and the risk of developing new antibiotics is also a business model for how far and how expensive it is to develop new. So why should we search in the Arctic marine environment? So most of it's largely unexplored, and marine microorganisms in particular. As you mentioned, penicillin was discovered by humans, and that was from terrestrial fungi. So a lot of the friendly microorganisms that he referred to earlier, they are actually the producer of many of the antibiotics, or the precursors of the antibiotics that we use today. So we are thinking that the marine microorganisms is the new source that we should explore, because they potentially have new antibiotics molecules within them. And we also think, adding with the Arctic, that these organisms have developed different strategies and they have a different approach, and that's why they might have a bigger chemical diversity that we could tap into. So DigiBiotics, the project that are part of the Center of Digital Life, it is a multidisciplinary consortium consisting of 6 work packages that are aiming to find new antibiotics and develop state-of-the-art tools to study peptide membrane interactions. So the goal is to find new antibiotics that have less chances of having immediate resistance development. So membrane acting that are targeting the bacterial membrane is in the focus. So I will walk you through the project because it's many work packages. So work package 1, where I'm in, is biodiscovery and using the marine microorganism to find new chemistry that have antimicrobial properties. Work package 2 is organic synthesis, making the natural products, but also synthesizing antimicrobial peptides to develop further. Then we have work package 3, which is optical spectroscopy, but also working on the theoretical approaches on how these natural products that we could develop further and build out tools to better understand the interaction of these new compounds and how they work. And work package 4, the spectroscopy here, also the structural elucidation. Sometimes the natural products compounds, they are quite complex and diverse, so it requires a lot of expertise to just solve the chemical structures. But also here, studying membrane mimics is also part of this work package. So you see, it's quite heavily chemically loaded projects. And here also the dynamic simulations, drug membrane interactions is part of the DigiBiotics to how can we use and feed back with information from the new things that are discovered, develop tools, etc., to study this even better. How can we find new antimicrobial compounds? But then again, linking again back to the whole project is connecting this using clinical isolates that are resistant, so that we are not only using lab strains to test that we have activity, but also see that the compounds we are discovering do have proper activity also against what we call the bad bugs. So you see it's a big project, so I will try to walk you through some of the processes that we do, especially here in work package 1, and where do we start with the discovering this marine microorganism. And so the platform where I'm working on, we are trying to identify the novel compounds. We are bringing in the marine extracts, try to purify chemically, test them for biological activity, and identifying this. And usually you see a pipeline presented, but it's actually going in circles because we use this bioactivity, the antibacterial activity, to guide us to which compounds or which samples that we are studying. And then hopefully have a lot of active compounds coming out of this. But now, since we have the DigiBiotics and the marine microbes, where we actually can sequence the genomes, where we can start accessing not only like the physical chemical extracts, but actually also looking at the genome potential, we can study the biosynthetic gene clusters and to see who are the most promising, who can probably produce interesting compounds. But anyway, they have to tap into, for us to be able to test them in the lab, they have to go through and be expressed so that you actually can test the active compound in the end. So how can we explore the marine microbes? Yes, as I just mentioned, we could use bioacid guided isolation, but using unique marine microorganisms. We see sometimes that we have to trigger them to produce because they can be silent. So we might have to co-cultivate marine fungi and bacteria to try to trigger them to start producing interesting molecules. But also this genome-enabled biodiscovery, which DigiBiotics is all about. Like, how can we start mining with all the genomes now coming up? We mine them for biosynthetic pathways for these interesting chemical compounds, these secondary metabolites, and try to use this information together with this bioacid guided isolation to increase the chances of finding new compounds. So we are lucky to have a very unique collection of samples. So we have access to a lot of marine bacteria that are coming from sediments, seawaters, and macroorganisms in the sea. And we have more than 600 species that are in the freezers here. Some have been genome sequenced and some are waiting to be that. And we have a unique collection of marine fungi that are isolated from driftwood, sediments, and algae. And here the numbers are increasing, 250 species here. And how the one that we actually can grow in the lab, we are fermenting them into these liquid cultures to produce biomass so that we can start extracting chemistry out of them. But there is a challenge. These marine bacteria and fungi, to have them in the lab is not just straightforward, because we see that there are much more of them out there, but it's not easy to make them happy in the lab and actually start producing the chemistry that we are looking into. So that's part of the challenge in working with these marine microbes. So just walking you simply through. So when we scale them up in this liquid culture, we are, after a while, and we have enough biomass, we are adding this resin that we're trying to extract the chemistry. So usually you could say that we get a chemical extract out of the flasks, but sometimes they are too complex to start screening. We have too many molecules present at the same time. So what we do is trying to reduce this. So before we do the screening, the antibacterial screening, we try to reduce the complexity by fractionating them and going through this pipeline, as I presented this in the circle, to try to dig out and guide us through to either, do we find a novel structure? Do we find a novel bioactivity? In the end, working through each of these bottles that you saw in the previous picture. So the lab strains that we normally test in the initial part of the pipeline is that we do gram-negative and gram-positive bacteria, but we also do inhibition of biofilm formation in the lab to guide us to which fractions extracts are of interest to further pursue in the pipeline. So, and what have we worked with? This is one of the examples that I wanted to bring up in the DigiBiotics-project. This is a fungi that was isolated up outside on the coast of Berlevåg, up in the north. It was actually in the splash zone from a dead pine. This was Teppo Rama that was working on looking for new marine fungi. And he characterized this to a family, Lulvortiaceae family. He brought it into the lab, and actually one of the PhD students in DigiBiotics started working with this fungi. And I have to say, this was really slow growing. It took 40 days to just get it happy on the plate, sufficient amount, to bring it over to the flasks here. And I think it took more than 100 days to actually produce sufficient biomass to get an extract out of this. But the patience of Marta growing this in the lab turned out to be good, because when she tested the fractions against Staph aureus here, you can see there are 5 fractions. This is inhibition of bacterial growth. And you can see here in fraction 5, we actually have a hit against the gram-positive bacteria. So it was worthwhile waiting for that slow-growing fungi. And it was a long process. And then all the chemical work started to dig out what's in this, producing this antibacterial activity. And this was published last year. It turned out to be a new compound, a new dimeric naftopyrone that are coming from this family. It's actually the first metabolite isolated from this family at all, but I can understand because it's quite slow growing to work with. But it turned out to be active also against coupling on work package 6 with Erik Juskiewicz here, that it also had effect on this clinical resistant isolate. So it's not only was active against the lab strains, because we want to translate this into further molecules. So this proving that we can find new things from the sea that has antibacterial activity. But anyway, this is just the starting point of developing things. There are a lot of things that needs to happen, but we have to look, and we can dig out new chemistry that have really good activity from this natural points before we start optimizing activity. Before you start tweaking around, you have quite good activity here already. So coming back to this DigiBiotics project, so we have in work package 1 coupled this through the different work packages where all of them have contributed with either structure elucidation, but also the bad bugs testing here in work package 6. So I think we started off with very different topics, but we have been able to merge and work together throughout many of these work packages. Unfortunately, there are many interesting results from many of these, but I don't have time to dig through of them, but I would just encourage you to look up more. And there are a lot of good research coming out of all of these work packages. And I could just mention, these are all the people here. And the 2 of the latest papers I'm just mentioning here is a follow-up on the mode of action of the slulvortinone compound that came out this year. But there is also a method developed how to study membrane bacteria, like how can we use model system to study membrane barriers, which we think is an important target for antibacterial testing and how to understand to have less resistance. So here, Jon Sigurd Svensson is the project leader of this DigiBiotics, and you see there's a lot of names here, many people involved, but it's been super to work with them because we have to learn from different research approaches. But I hope I have showed you that it's possible to find antibacterial compound in the marine environment, in the Arctic marine ocean. And summing up here, I see that time is moving. This is sort of the part where we have been contributing the marine microbes. We have now been able to whole genome sequence 5 fungi, marine fungi, and we are increasing the numbers on the bacteria also. So we hope to even digitally also contribute to profile which one to study in the next phase, which one should we bother to do the fermentation, bring them all through the lab and through the testing here. Because in the end, we want to isolate and feed the pipeline with new chemistry so that perhaps working in a new way, so new mode of actions which is needed for new antibiotics, that somebody could pick these up and develop them further. So there is a need for new antibiotics as part of the solution. And where should we look? So I'm just using Dr. Sanjay Gupta, that is the medical responsible at CNN. He was with us out on a cruise, and he said, go to the Arctic and look for new antibiotics. So thank you. Just keep following that advice.